Trout's eye contains 4 types of cone cells. The human eye contains 3. The fourth cone, with a sensitivity peak at 360 nanometers, sits below the threshold of human vision entirely. It detects ultraviolet light, and what it does with that information changes depending on how old the fish is. Humans are trichromats, meaning we build color from 3 cone inputs covering blue, green, and red wavelengths. Trout are tetrachromats. The UV cone is concentrated in the dorsotemporal region of the juvenile retina, positioned to sample light coming up from below, exactly where prey in the water column would appear when viewed against open sky. And this isn't coincidence. Zooplankton are nearly transparent in visible light, but reflect ultraviolet light differentially. Research by Professor Navales demonstrated conclusively that juvenile rainbow trout with functioning UV cones located zooplankton prey at greater distances and across a wider field of detection than fish without them. The cone exists to find food that is otherwise invisible. Studies measuring UV sensitivity directly from rainbow trout optic nerves found a UV cone mechanism present in fish under 30 grams and no detectable UV sensitivity in fish over 60 grams. The specialized cells are called accessory corner cones, and they progressively disappear from the retinal mosaic as the fish grows. The juvenile visual system and the adult visual system aren't the same instrument. What adults retain is different but equally worth understanding. At the level of the torus semicircularis, a brain region responsible for orientation and detection, UV-sensitive neurons persist in larger fish even after peripheral retinal sensitivity diminishes. These neurons are specifically linked to polarized light detection. Rainbow trout process the polarization angle of incoming light across multiple orientations. During crepuscular hours, when the sky's polarization pattern is strongest and UV penetration in the water column is near its daily peak, this system appears to function as a spatial reference, potentially aiding orientation and prey detection in ways that visible light vision alone can't provide. The practical implication for anglers is genuinely unresolved. UV reflective fly tying materials are sold on the premise that adult trout detect UV differences in flies. The science on adult UV sensitivity makes that premise complicated. UV light penetrates clear water well and murky water poorly, and it's effectively absent beyond 1 meter in stained conditions. The fish being targeted, the water being fished, and the depth of the presentation all determine whether UV properties mean anything to the animal on the other end. The juvenile trout hunting zooplankton and the adult trout tracking a fly aren't running the same visual system. Both are seeing something the angler can't. What exactly that is depends entirely on that fish and the circumstances.